Okay. Hi, everyone. My name is Igor, and I'll be instructor for the single cell workshop. Okay. So we'll have 2 days, today and tomorrow. So basically, today we'll talk about like some theoretical aspects of single cell. Like basically, we'll talk about technologies, methods, what you can do with this data, what you should expect, what kind of analysis you can do. And tomorrow we'll talk about, it will be more interactive course. So for tomorrow, I'm expecting you to install RStudio and some other stuff. I may come up with this later and send you in an email. Okay. So first of all, I'm expecting that all of you are like from biological background, more or less, right? I'm from computer science, from computational part. But first of all, because we also have some here, we have some graduate students, right? And for like our purposes of how we teach this class, I would like to invite you to basically go to this link. Okay, and there is some very small quiz. So let's assess our knowledge of single cell RNA-seq before this workshop and after it. Okay, so I should have sent it in an email. I don't know, like if you guys got the email from me yesterday about this workshop, I can send there, okay? So let me send it. What happens? Okay, reply to all. Yes, in a couple of seconds, you're expected to have the link. So I'm showing you what should happen. This is something like this. All right, so what you should do is just type in your email address there. It's very important for you to type your actual email address because you'll get all your answers, and this is for our purposes to track how you answer the questions. So there are only 10 questions, so let's dedicate maybe 5 minutes at most. Okay, now I see. Okay, you have more background. Okay, well, let's go through the questions. So which of these is not a single cell technology? Basically not single cell RNA-seq technology. Okay, so there are 4 answers and you should choose one of them. Okay, please don't tell the answer. So next, which single cell technology is more sensitive? It detects more genes. Like which one, SMART-seq2 or DROP-seq? Then when a gene is expressed but we don't observe any reads, we call it one of these 4. So basically, the next question is what UMI stands for. There are 4 answers. What is a doublet? Okay. So basically, a question: how do we measure gene expression? Which kind of what units do we use? There are 4 answers for this. And then when we want to transform the data, let's say we have single cell data. So if we want to reduce the impact of highly expressed genes, right? So what do we do? It's one of these 4. Then, for example, if you want to identify cell types with single cell RNA-seq data, there is a procedure. So it's one of these 4. Question 9 is which technology is cheaper? Basically, let's say per cell, right? Okay, is there something funny? What? But cheaper is a comparison. Out of these 2, one should be cheaper. All right. What?
So she was wondering about how come I didn't get the material. You signed up 2 hours ago. That's why.
Oh.
So that's what happens when there's no meetings like deadlines.
Okay, and the last question would be, what's the main tool for single cell RNA-seq analysis? There are 4 answers. So did you do all of them? Yeah. Answers? Oh, great. Okay. Yeah, so please answer these questions. If you don't know, or just guess an answer out of these 4. But otherwise, you know, like after the class, we'll see how your results improve. So if you are all done, let's begin the slides. Done? Done. But please submit it now and not don't wait when you find out the answers during the workshop. Okay. Oh, I have 18 responses. How many people have? No, someone is still huh? Yeah, this is how we usually do this workshop, because like in the first day we just talk. In the second day we like do the actual analysis. You will run some R tools, you know, and that's usually enough. Yeah, no third day. Yeah, I mean we could have, you know, like a weekend, you know, it would be a lot, but I think one day like theory, one day practice is usually what we do. Okay, so let's begin the class. All right. Okay, so this is single cell RNA-seq workshop. The outline is the following. We'll talk about single cell as an alternative to bulk RNA-seq. So we'll talk about what is this, what we can do with it, what are the technologies, because when we talk about single cell, it's not only one technology. You know, it's a whole bunch of technologies. We'll talk about some of them. In particular, we'll pay our attention to SMART-seq2 protocol and 10x genomics. So this will be 2 things to consider here. And then basically when you have this data, when you apply these technologies, what is the output? So what do you see there? How do you get the information from this technologies? And then like the most interesting things would be, okay, from computational point of view, would be how to process this data and extract some meaningful information. Okay, so basically we'll study how to do normalization, how to visualize single cell, at least like from, you know, from theoretical point of view, more or less. And then what is differential expression? How do we identify cell types? What is a cell type in single cell? And we'll briefly go over software tools and data sets and some kind of ideal single cell pipeline, which is a kind of weird word here, right? Ideal, because it's not ideal for all type of analysis. It depends on what you want to do, what kind of data sets you have, what is your budget, a lot of things, right? Okay, so probably the most interesting would be, you know, if you have your data, I assume most of you would like to find out what's the ideal way of using it. So let's go over the steps, and maybe at the end we'll see at least some some answers. Okay. So first of all, single cell versus bulk, right? So basically, people are used to do bulk RNA-seq, right? It means that you, for example, you want to profile a tissue, but have like, let's say blood, right? Blood is a mixture of cells, what happens is that you take this mixture, you basically sequence all the cells in that mixture, which gives you only a point estimate of mean gene expression, average gene expression, right? So basically, for you have multiple millions of cells there. What happens? There is a whole soup of the cells. You extract RNA, right? You sequence, and you get only one number per gene in that sample. So it's kind of already we can tell that it's an old technology, right? And it's relatively cheap because people, again, we can discuss a lot, some philosophical discussions on how cheap or expensive it is, but it's cheap compared to single-cell RNA-seq, which is a newer set of technologies. And one of the most important representatives are SMART-seq2 or let's say 10x. So it appeared only in 2009. It's kind of also old, but it got more attention since 2014 when such technologies as SMART-seq or 10x appeared, and now we can profile in one experiment up to 1 million cells. And this number is expected to grow. So what's the advantage of it is that it allows us to capture the distribution of gene expression across cells. So it means that we can literally profile each cell separately instead of coming up with a number for gene expression across many cells. So the downside is more expensive. Okay. It's more expensive because it's more basically, let's say, library prep steps, isolation, all that stuff. It's more expensive, but okay, the advantage is it's more powerful, right? Because we can do many things which are not able to do with standard bulk RNA-seq, like cell types, right? For example, with single cell, we don't necessarily need to do laborious, let's say, FACS sorting or some other things, right? To separate cells into cell types. We can do this computationally, and then do all standard transcriptomic analysis based on cell types. Because nowadays everyone is thinking that the biology is is kind of cell type driven. So many things happen on the level of cell types. We can do, for example, cell trajectories or cell types. Identify the development, many things. So it's more powerful and it allows to answer more biological questions than bulk RNA-seq. So another characteristics is that the data sets range from, let's say, 100 cells, but now it's already in the past. Now we are approaching to 1 million, so 10 to the power 6, 1 million cells, and and this data sets increase, and basically the challenge is having this large data sets is to understand what's going on there and how to what's the biology there. Okay. How to go to the next slide? Okay. So what is single cell? Okay. Again, this is a bunch of technologies, and here, if you see 2009, it was I think 8 cells. In the very first single cell paper, we only could observe only 8 cells, right? And as you see, as the time passes by, you'll see that basically the throughput, or this number of single cells in a study, is growing. So now it's already 2019, right? And we're approaching 1 million here. And for example, 10x genomics, this is what we'll talk about. It's one of the choices nowadays. Okay. Okay, there are some other technologies probably which are newer, but people are not using them that much as the well-established ones, such as 10x genomics. Okay. So what's the, let's say, RNA-seq workflow? Okay, if we have, for example, some tissue, let's say we have a solid tissue, right? And what do we do with it? So basically, we take the solid tissue, and first of all, we need to dissociate, right? Basically, to separate cells, to have separate and then isolate. We have dissociation and then isolation here, right? So basically, we want to have separate each cells and not having, again, a bunch of cells physically. What do we do next? We extract RNA, and then the usual thing in sequencing. So this part is basically sequencing here. So what do? We take RNA and then we convert it to cDNA, right? And then we sequence the cDNA with, for example, amplify with, for example, PCR or RT, reverse transcription. Then we amplify, right? Amplify cDNA, we sequence. So this is whatever, like some Illumina or some other sequencing methods here. And then after we sequence, we get file with FASTQ file with reads. Okay, so we look at this reads, and what we do next is, okay, if we have bulk RNA-seq, that's it, right? We quantify, we basically align these reads to the genome. We see how many reads come from a particular gene, and basically, for example, a differential expression analysis follows. But with single cell, what we do is basically a bit different. So we have to, we get single cell expression profiles, which is a gene expression matrix, right? So basically here for each cell, like, for example, rows here are cells or genes in this case, and for example, columns will be cells. And we need to understand what's the expression of a gene in a cell, right? So we'll get single cell expression profiles. We do clustering in order to find out, groups or populations of cells, and then identify cell types. And what happens? We do all our biology, all our experiments per cell type, right? Or we, for example, want to understand the developmental stages of cell types. Many things. Okay. So what is single cell technologies from the perspective of wet lab, right? So basically, there are different, for example, methods like, let's say, microfluidic chips, right? So there are like technologies as SmartSeq, SmartSeq2. So this is one of the types, and then the other type. So they're also called like plate-based methods, right? So the other one is basically droplet-based. So on droplet-based, the idea is that we isolate each cell in a droplet, and we do all, library prep there, right? We basically prepare the library, and then we sequence with usual Illumina technology, right? So basically, here the first step would be cell isolation, right? And here each cell is loaded into a well, in case of this microfluidic chips. So what happens next is we amplify. Right, for example, amplify the RNA with PCR amplification, right? Here, amplification is done individually in each cell. So here, after we, the PCR amplification is, is pulled when we have all the cells there. After we prepare the library droplet, we pull them and do PCR amplification. And then this is the, the common thing between these two technologies is, or like two sets of technologies, is that we do sequencing here, right? And then we do all kind of applications. So one of the important things here is difference between droplet-based methods and microfluidic-based is that in droplet-based we usually do 3' end sequencing. So it's basically we have two transcripts, right? We only sequence the 3' end here. Whereas in microfluidic ones, we can deliver full-length sequencing. So it means that we can distinguish isoforms. Okay, is this distinction clear? Right? Yes, 10x is also here. It's droplet-based. So it means that you can quantify, on the gene level, but not on the isoform level. But here with like SmartSeq or SmartSeq2, right? You can understand gene expression on the level of isoforms. In case you need in some applications, you might need to answer this question. So what's the gene expression of several isoforms? Or if you want to detect a novel isoform, you will not be able to do this with 10x. Okay, I think the question might come from the previous steps, like amplification. I'm not really sure, but I think it's because you do pooled PCR, right? And probably it depends on how you tag your molecules. It might be the case, right? In 10x? No, you have ribosomal, mitochondrial, everything, but oh, I see. Yeah, we'll talk about the library prep steps in the next slide. That's just acknowledge this is one of my weakest points of mine in this slide. So you guys know this biology better than me. But I assume that can be explained by the fact how we prepare this. Less what? Yes, exactly. We'll go into, we'll explore this characteristic later. But that's all right. With 10x, you can get, like millions. Okay. So this was wet lab stuff, right? And for example, with computational, like as we get the data, right, from single cell, what do we do next? Is, for example, the first step would be QC or quality control, right? You'll need to, so what you'll get, we already talked, right? This will be basically a matrix where you have cells and genes, and you know like how much every gene is expressed in each cell. So basically one of the first steps will be QC. Here by QC, we can understand, for example, the simple procedure of filtering out cells and genes, which might be, spurious for some reason. Let's say a cell might have, only like 100 genes expressed. Most probably you would like to remove it from your downstream analysis. Or it might be that, like some genes are expressed only in 2 or 3 cells. In such case, it might be an interesting gene, right? But most probably you'll not be able to do any conclusions with such a rare gene. So usually this is the steps we do in QC. Then, for example, we do read alignment. In 10x, these all things are done in their software. In SmartSeq2, you basically get reads for each cell, and you will need to do read alignment, then quantification, right? For example, next, you can do imputation. This is basically inferring missing values. Like for example, some because technologies are not perfect. What can happen is that we can lose some of the values, in the matrix. So although we see that like a gene might be expressed, but we see 0 there, right? So for these cases, we would use some kind of imputation, and then we do cell type identification. Basically, we can do differential expression analysis between the cell types, right, to understand what are the marker genes for each cell type. Or we can do differential expression analysis between different conditions. This is less used. Okay, and then we can do any other things. For example, we can do on cell type level. For example, we can explore like what are the upregulated genes from the perspective of gene ontology, or we can explore, for example, immune repertoire. If we catch, we might catch something from that region, like CDR3 region, understand what's the immune repertoire. Or we can do association testing. Let's say if we also have the genotypes. Right? If you have the genotypes, you can do, for example, some kind of like single cell EQTL analysis, right? On cell type level. So many things. Okay. Now applications. Multiple applications in developmental biology. So we said that we can basically understand, how different cell types are connected to each other, and this is called usually like pseudotime analysis. So basically, you want to understand how your cell types evolve, in developmental order. So this would be one of the things. You can cancer biology. Okay, mostly it's it's dealing with maybe DNA and not RNA, but okay, I included it as an application of single cell technologies. And you can do it like, for example, neurology, right? So, for example, here, discovering mechanism of cellular development. Mentioned this for many times. We can identify gene expression signature across different cell types. It means that identifying which genes are upregulated, let's say in one of the cell types, right? And then we can characterize functional differences among the cells of the same cell type. You know, like some cells might be in some states compared to the other ones. So for example, some cell cycles or some other things, like many things, you can do it with single cell. So cancer biology, right? We can like hypothesize about different cancer models and then find evidences with it. We can, okay, these two things, the second thing, again, it relates more to DNA-seq, but you know, just for completeness here. And neurology, you can explore, let's say, okay, genomes, and not only genomes, but also transcriptomes of individual neurons, right? For example, you can see how they, you know, what's the response on different, you know, stimulations and so forth. Measure gene expression in, you know, in individual neurons, and then identify which genes respond to some conditions. So, okay, many things. So multiple applications. It would be interesting to know what are your applications, because I assume that if you came to this workshop, then you or your PI probably have somewhere, you know, a single cell data set, and you would like to analyze it. Okay, so it might be something which is not listed here. Okay, this is single cell technologies. This is one of my weakest understanding, like literally, because you know I have never done any web lab. So, but let's talk briefly about the technologies here. Then I don't see. I'm not sure if you see all the pictures here. So basically, here we talk about the following: that there are multiple ones, right? We already mentioned like DropSeq, SmartSeq, right? So, for example, here we see like SmartSeq 2, right? Okay, like SmartSeq, MarSeq, DropSeq. So here we have like 5 technologies listed, right? And basically there are some differences in how they, you know, for example, how they construct the library, right? So for example, some of them like CellSeq and SmartSeq, right, is like plate-based, and the other ones are basically droplet-based, right? So nevertheless, the next step in all of these methods is reverse transcription, right? Because basically when you look at the RNA from single cell, right, you would like to, right, to make it cDNA, right? And then basically what happens, you want to amplify, do cDNA amplification here at this step, right? Or like some of the protocols, they also require a second strand synthesis, right? And then there is library construction, after which we get the reads. So here it depends on which technologies we use. The reads, you know, or like this is, what is a read? A read is basically, you know, like a small region which we sequence from a fragment of RNA, right? So depending on the technologies, we'll get different reads. So what I mean by different reads is that, for example, some of the technologies, they attach some tags, like for example, sample tags or molecule tags, you know. So based on this, we will distinguish technologies as, for example, like here in this slide, we're distinguishing the technologies based on 2 criteria. The first one is basically whether they are UMI based or not, and if they're full length or not. For full length, it's already clear. We talked about full length. Full length is when we can generate full length transcripts, right? So for SmartSeq 2, we have full length, right? For SmartSeq 1 or what else? So basically, it's only SmartSeq, right? For SmartSeq, we can generate full length transcripts, whereas for all other technologies, you know, it's not full length. It's only like, for example, either 5' end or 3' end. So for example, for 10x, it's 3', right? Another thing is UMI. UMI is basically, it stands for unique molecular identifier, right? So hopefully you chose the correct answer. So unique molecular identifier means that there is a tag attached to the molecule, so that, like to the fragment, we sequence it, and we need it just for the fact that, you know, when we amplify, the amplification process is not uniform, and as a result, it might skew, you know, the gene expression which we see, like the read counts which we measure. So in order to remove the amplification bias, we use this UMIs. These are special tags, which allows us to, track whether two reads come from the same molecule. Okay, we'll have a slide on that. So here, sensitivity. So different technologies produce different results, right? So this is one of the things which you should consider which technologies to use in your experiment. So for example, in Zhang et al. Paper in 2018, right? So there was a comparison of different technologies here. So I assume that you should see it, but if you don't see it, I'll send the slides afterwards. So here we look at different technologies, right? And there are some plots from which characterize them from different aspects. So for example, in the first one, we see on the x-axis sequencing depth, right? And then the y-axis is number of genes. So basically here, what it means is the following. If you sequence more, right? If you sequence, probably you don't see it, right? But I'm telling you. So this is sequencing depth, and this is how many genes we detect. Obviously, if we increase sequencing depth, we will detect more genes. So for example, if a gene is less expressed, right, it's less chance that you will catch it with less reads. But as you increase sequencing depth, like this line, basically how many genes we detect, it kind of stabilizes and it saturates at some moment, right? So the main, idea here is that SMART-Seq2 is basically captures like almost everything. So as you increase, for example, sequencing 10 million reads per cell, right? Or if it's not, we can go to this paper and look at it. So as we increase depth, we see that, okay, we can, from individual cell, we can see more genes, right? As compared to other technologies as DropSeq, which is much less genes, like, how many, let's say 5,000. For DropSeq is 5,000, right, on average. And for SMART-Seq2 is 8,000 genes per cell. Okay, so the second one is showing basically the distribution, right? Like this is the same picture as here, but it shows us like different technologies. So for example, this is CellSeq, this is DropSeq. So the distribution of genes as we see. So for example, the average would be here, like how many, like 6,000, right? So here will be 8,000. For SMART-Seq2, it will be much more. Right? This is the distribution of genes which we detect in one cell. So next, the third one is basically how many genes we detect if we if we alter the number of cells. Okay, so if we consider, for example, 100 cells, obviously we'll catch more genes. It probably also depends on the biology, because sometimes we might not catch a gene because it's biologically it's not expressed in the cell. So it's kind of, anyway, it's a kind of stochastic measure. But on average, we see that SMART-Seq2 is also better on this thing. So basically, we take less cells because of the fact that we detect more genes usually, like here, right? We will detect more genes as a function of cells. And it's also better compared to DropSeq, right? Okay. So this is the distribution of this thing, like how many genes, right? Okay, so what are the issues with single cell data? It has a lot of issues. So despite the fact that it's kind of advertised a lot, people are switching to using single cell more and more, but it has many issues. Like the first one, this is kind of continuation of the ideas from the previous slide. So if we have like a good technology, right, we would expect to see a gene expressed. If you know that a gene is expressed in a tissue, right, we would see this gene expressed in all the cells. Right, so we would expect something like this. This would be the distribution of gene expression across all the cells. Right, but it happens, in fact, in single cell that in many cells we don't observe a gene. which is supposed to be there. Okay, so we call this thing a dropout. So single cell is known to have a lot of dropouts. So what it means that the gene expression matrix, which you get from your single cell experiments, is very sparse, right? Okay, so this is one of the issues with single cell, right? So another thing is like an issue which is characteristic to, like let's say to SmartSeq data, because you have PCR, right? You might have amplification bias. It means that some of this, some of the reads will get amplified like by, let's say, 1,000 times, right? And some reads are only like 2-3 times. So you will see a big difference between those ones. And technologies which use UMI, okay, we already see that this is, for example, 10x, right? They attach a specific sequence to each of the reads in order to, see whether reads comes from the same physical molecule. Okay. Oh, sorry, I think it's detecting. Yeah. Okay, this is a small break. Yeah, we'll talk about this. We'll have slides on this. I think it's maybe too much time. Should find it. Yeah, let's see what we can do, or maybe. Okay. Yeah, sorry guys, this is a technical problem. Okay, maybe let's check. Oh no, everything is fine.
Yeah, so the other one with black board that you made, just this young one, very sensitive.
Yeah, I
so
think
you budge this, it's gonna.
Yeah, I'll be more careful. But for now, it's okay. Yeah, thank you. Okay. Small break. Okay, what's the other issue with single cell, right? Let's say you have like 1 million cells, right? Usually you cannot, or like still in many cases, you cannot sequence them as one batch or one experiment. So what people do, usually they split their cells into many experiments, technical experiments, right? And then what happens when they analyze the data, right? Let's say they plot the cells. We'll discuss how to plot them, like how to visualize. But when you visualize, you see that there is some difference. Although you might have some, let's say, some cells which come from the same biological condition, right? But because of the technical artifact, you'll see that there is some difference between the experiments. So we call this differences as batch effects. So for example, you can have batch 1 and batch 2. When you visualize the gene expression matrix, you'll see that, for example, some genes are more expressed on average, like all of them, more expressed than the genes in the other batch. So although, you're expecting to see the same thing, but because of the fact how many effects are there, like technical ones, or like let's say in the lab someone messed up something, or like many things, or like stochasticity of your experiments. So everything results in this artificial differences between two conditions. So these things are called batch effects. Okay. SmartSeq2 protocol. Okay, this is how library preparation works here, right? It's like a SmartSeq2. So basically, First step would be cell lysis. Right? We take the cell, we break it down, and then what happens? We extract poly-A RNA. That's what we discussed at the beginning. So you have poly-A RNA, right? You basically attach some primers there, and then what happens is that you do template switching, you do basic amplification, right? And then one of the steps here would be tagmentation. So basically adding some tags, right? So which results in reads having, like, for example, some primers. So this will be some primers, right? Which should be attached to the reads I5 index. So this will be like sample indexes, right? So we will understand which cell this read is coming from.
So what was your problem? Can you not normalize out the application bias? You pull this transfer descendants.
Can we normalize?
Not normalize out the application bias. You pull this transfer descendants.
Can you not normalize?
Yeah.
I don't really understand the sense of this question. Why wouldn't you normalize if you, okay, if you want to remove all the confounders? Okay, maybe we will reach at some point where this question will be answered. So, okay. So this was, the way how library prep is done. But what's the take-home message from SmartSeq 2 protocol is that it's basically it has, despite the fact that it's more expensive than, for example, droplet-based technologies, but it has some really nice properties as that you don't need minimum number of cells for experiments, right? So basically, if you have like 5 cells, you can sequence 5 cells. So basically, it does not require some specific amount of cells to be loaded there. 100% capture rate. So basically, all cells are assayed. Compared to 10x, in 10x, basically what happens is as far as I remember, it's around like 57% of cells which you assay. In fact, like you load, for example, 100 cells, and you only will get like around 57 on average, right? So compared to that one, so here everything is assayed. And then, okay, it's a bit like it's a bit exaggerated, but I think it's that okay, we can say that every transcript is detected. Okay, based on the previous pictures, we saw that it detects more, more transcripts and genes, right? And then we have full-length transcript sequencing, which again is useful in, for example, novel transcripts, right? Sometimes you have novel transcripts, you can detect them with SmartSeq 2 and not with other technologies. There are no doublets. Basically, okay, what it means, each cell is, is basically is not mixed with some other cells. So basically we isolate and we put the cells into wells, and we don't have, multiple cells sequenced together. Yeah, drawback that it's not a cost-effective option, right? You spend more money, but it's easy to use and it's open source. So all the tools are, open source. Like they basically you can use them, modify if you want, all the software.
How many cells? The typical assay for SmartSeq 2, how many cells can you do?
Okay, I would say that it's a couple of thousand. Right? This is like the data set, usual data sets is maybe like 2,000-3,000 cells, something like that. But I think it depends on how you on the sequencing, right? So this is the sequencing step. If I correctly understand the question. So the SmartSeq is basically you sequence like both SmartSeq and both 10x. You like one of the steps is Illumina sequencing, right? If you're using stranded protocol, then you keep this information, right? It depends on which sequencing you're using, right? Yeah. As far as I know, that people are using now stranded protocols. Right? So it depends on how you sequence, but you know, basically the differences we saw already. The differences between different technologies is the way how you library prep, right? For example, in 10x, you do droplet based. In each droplet you do like, all kind of things like emulsion PCR. All these things are done inside of each droplet, right? And then you do like, let's say, full PCR, right? And then you sequence. And for example, here you do each cell separately. So, but then it depends on how you sequence. Like this information is, you know, either you retain it or not. It depends on the sequencing step. Okay, 10x genomics. So let's talk about UMIs. So what is UMIs? Because usually we deal with UMI when we deal with 10x. Right, so basically, if we look at a read, let's say we have a read here, right? It has, first of all, it has self barcode. What it means? It means this barcode is basically, I think its length is somewhere 16 base pairs, right? So what it means that if we look at two reads and we see that this cell barcode is the same, it means that they come from the same cell. And then this UMI, this is another sequence which is a part of a read, which tells us whether two reads come from the same molecule, right? Because what can happen, let's say we have a gene, all right, which expresses physically some transcripts. So these transcripts would be different, all right? And this means that basically the cell barcode will be the same because two transcripts come from the same cell, from the same gene. Right, but UMIs will be different if they come from different transcripts. And then the other part of the read is cDNA. This is the useful information which we need. So these two things, like cell barcode and UMI, we need to to classify reads. So which cell it comes from and which transcript.
So the UMI comes first, and then the cell barcode.
Oh, your question is.
Well, my question is how do we separate between cell by cell?
Oh, between cells?
Yes. So that's from the UMI.
No, okay. This is from cell barcode.
Okay.
Yes. So this is okay. So for example, when you look at the read. So this part will be cell barcode, right? So for example, I have a cell 1, and this will be cell 2, right? And suppose I have here one gene and here one gene. Right? So what happens, let's say we observe like some reads here. This is UMI. Let's say this gene was expressed twice and it produced 2 transcripts. Right? What happens is that this is a read from this transcript, this is a read from this transcript. And the same here. Right? The same, let's say we have 2 transcripts floating in this cell. Right? This is one gene. It's a simple cells with one gene. Okay, what happens? So, okay, this is here, this is here. So this is cell barcodes, this is UMI, right? What we will see that these 2 reads, because they come from cell C1, they will have the same cell barcode, right? And then these 2 reads, because they come from cell C2, they will have different cell barcodes, okay? By looking at the cell barcodes, we'll understand whether they come from the same cell. And then here next, we will have UMI, which is, I think, is from 8 to 12 base pairs, something like that. Okay, which allows us to distinguish whether these, whether these reads come from the same physical molecule. So what happens? We might have one more read here, right, which will have the same cell barcode. But what can happen here is that UMI, let's say this UMI indicates that this read came from this molecule. So next. Do I have some other colors? So let's say this UMI indicates that this read came from this molecule. And what happens if we have another read, let's say we had some amplification, right? So we have another read from the same molecule, right? So basically we will look at it, and in some other circumstances, let's say when we don't have UMI, right? We would see, okay, we have 2 reads from this molecule. But this is amplification bias. So we can reduce it by looking at these 2 and clump them together and say, okay, this is actually like one molecule. So yes, in 10x, what happens, the number which we see for each gene and the cell is basically, it's telling us how many physical molecules we have, because we can clump the reads together into 1 and treat these 2 as 1, because it's in fact, you know, 1 UMI. So that's why you like for 10x, you might hear that it's either read count or UMI count. You know, so basically we're counting molecules with 10x. Okay. Did I answer?
Yes.
Yeah. Good. Okay. Adjust the camera. Oh, okay. Yes. So I was talking about different cells. If you have different cells, right? So what we can have different reads coming from different physical molecules, physical transcripts. And here, when we look at cell barcodes, we can distinguish whether two reads come from the same cell or not. And the second level of barcoding here is UMI. It can help us distinguish whether 2 reads come from the same molecule, as this case, or from a different one. So because of the fact that, you know, in sequencing, we might have sequencing errors. So what can happen is that, you know, there might be some error in UMIs when we sequence it, because it's also a sequence, right? We need to somehow read it. And when we read it with Illumina, let's say, it's, you know, it's not that likely, but still we can have some errors. And then what can happen is that we can misclassify some reads. You know, like, let's say, what can happen is a different UMI does not necessarily mean different molecule, right? So, okay, let's say what happens is that here in one of, like, these 2 UMIs are the same. What can happen is that, let's say we have some, you know, substitution here, right? Let's say we have, or like 2 substitutions, let's say. Okay, when we look at the sequences of these UMIs, we need to understand whether this is 1 UMI or 2 separate ones. So what would be, you know, the outcomes? So for example, if you treat this as 2 separate ones, right, you're kind of increasing the counts for, you know, for basically for the number of transcripts, because instead of saying that you have 2 transcripts, you will say that you have 3 transcripts, right? This is one of the possible outcomes. The other one, you basically need to understand somehow that these 2 are sequencing errors, and, you know, treat them as 1. So that's because of, let's say, sequencing error or PCR, right? You will see new UMIs. So how to understand that this is a, you know, artificial UMI or a real one? You, like, usually you somehow cluster them based on, you know, based on the sequence, and then see if it's, you know, if it's, for example, you see it only once, then it might mean that this is some error, sequencing error. Okay. What else can happen is that, for example, different transcript does not necessarily mean different molecule, right? So it might happen that we have, like, for example, mapping errors when we map the reads, right? We can map a read to, you know, to, for example, different genes, right? This is so-called multi-mapped reads. So it means that if we see different transcripts, it does not necessarily mean that we have different molecules. And then the third thing is that the same UMI like if you have the same UMI, let's say here, we don't see these errors, but we have the same UMI. It still doesn't give us 100% guarantee that this will be the same molecule, right? Again, because of the, for example, sequencing errors, or what can happen is, for example, bias in UMI frequency, short UMIs, right? Or like many things. It does not necessarily mean that in all the cases when we see two UMIs, this is like the same molecule. But again, like there are some like methods, computational methods, which can distinguish, like for example, filter out some spurious UMIs, or like decide when to clump them together or when to keep separate UMIs. Okay. But in general, like UMIs, they work okay. They really help reduce the amplification bias. Okay. So this is the slide which shows how 10x genomics works, right? So basically, we have 8-channel microfluidic chips, and then we have barcoded RNA. So basically, this is the whole pipeline, right? So we extract cell barcodes, UMI and RNA read. We correct for cell barcode sequence, right? Because again, we might have some error in cell barcodes and UMIs, right? We align the reads to the reference. Let's say we have human samples. We align them to human transcriptome using STAR, some of the methods. Right? Then we correct for UMIs, we count UMIs, and we produce gene barcode matrix, right? Produce gene barcode matrix. This is basically the whole pipeline of basically this is wet lab pipeline, right? And this is the computational pipeline for 10x. And if you remember, I was talking about like doublets, so that we don't have doublets in SmartSeq2. What it means that, for example, here when we have droplets, right? So in each droplet, we are expected to see only one cell, because it's a single cell. We want to separate cells. So usually, what happens is that some of the droplets, they will not have any cell. So they will basically have some ambient RNA, right? And then you will need to distinguish these droplets and don't take them into account. And then some of them, they will have like 2 cells here, right? Okay. And then what this picture tells us, it tells us that basically around 15% of the droplets will be empty, so they will not have any cell. So 5%, let's say, or 80% will have 1 cell. This is what we need. So this is good, and this is bad because we will have at least 2 cells in 1 droplet. So how can we distinguish such droplets? In many cases, it's easily distinguishable when you look at the total UMI count. Basically, it means that you collapse all the RNA from 2 cells into 1, and then you'll see a lot of transcripts coming from such cells. So you usually remove them from your analysis. Okay.
So what happened for the smart tubes? They do not have your count.
There you load each cell into a well, so you separate. You don't have this issue. And here is kind of automated because you have basically a flow of oil and cell reagents, right? So basically, and then these 2 flows are kind of mixed in one, basically in one place here, and then you basically get some cells which go into 1 droplet. So it's kind of automatic process.
So when they both have amplification bias, you have a little thing of water, could be a droplet, could be a well of a plate, with 1 cell in it. Then you still can amplify the DNA, cDNA.
Yes.
You do it as a pool, and 10x you do it individually in SmartSeq.
Yes.
Is that difference important, controlling amplification bias to the point where you don't need UMIs for
of difference. You mean difference in how you prepare the library?
Well, I mean, you use UMIs to control implication bias.
Mhm.
Are you do that in both SmartSeq and 10x?
No, just in 10x.
Just in 10x.
Yes.
So implication bias isn't as much of a concern in SmartSeq.
Yes, yes, but you can remove computation with this bias. You know, we'll look at it. Like you can do, for example, normalization. You know, like different things. But
Yeah, but you get unique reads from like
unique reads.
you can get uniqueness from like the length of the video.
Oh, like making reads longer.
Well, I mean, if you fragment it, it get longer.
Yeah, I think usually it's like the usual Illumina read length, which is, for example, for short read lengths is like, let's say 50 base pairs.
Yeah, but then you have you take a long piece, you take a long CDNA in the case of SmartSeq. You break it up into little bits. The part of the transcript that you see it on and the length of it gives you a certain indication of uniqueness. It's a two-dimensional, it's not as deep as a UMI, but it's a two-dimensional thing, both length and XY position on the transcript, whereas with three-time sequencing, you're always starting at the same place, so the reads are not, there's not a very good measure of uniqueness.
Okay, yeah, we will talk about how to, you know, like there are some units of measurement, like let's say FPKM or TPM, which take into account the length of the transcripts. That probably, you mean that, for example, longer genes produce more transcripts, right? And there is, for example, more amplification as a result.
If I have one molecule transcript, and I make a really long cDNA from it, and then chop it up with my Covaris, then, you know, I know what thing came from. I know that everything of length, 56, that's, at this position is probably all from the same molecule. Whereas if you have a very short, you have a very short cDNA that's only amplified at 3', a lot of them are going to be 56 long.
Yeah, usually when you measure gene expression, you account for the length of the gene. Yeah. Yes. No, for 10x you also have paired reads, where one read is basically, it consists of this barcodes, and then the second read is basically the useful information. That's what you have. Yeah, their website has a lot of
the barcoding thing is really a lot of stuff. So one whole read is dedicated just to the barcode, and then the whole other read is the assignment.
Yes.
So it's as if it was a single line, if you use pair density.
One is just for, like to understand cell and molecule. One whole read is spent for this. It's kind of single end, because one of the legs of the read is just information, like meta information, and then the other one is useful payload. Okay. So there might be, for example, with 10x, right, technical doublets. What it means that there is, for example, two cells are captured in a droplet, right? So it means that there are like really different cells, right? But the way how we isolate them, it does not, it does not dissociate them well, and as a result, we see two cells. No, basically this is due to technologies, right? So here, when we have this flow of cells and oil, and when we capture them in droplets, this is due to technology, right? Due to some artifacts here in this pipeline. So we might capture this two cells, and then, for example, we can overload the like, for example, microfluidic single cell experiment, right? We can have we would like to load it with more cells, right? And then as a result of this, we'll see more doublets. We'll see like a lot of doublets, and this is all like basically technical artifacts. Okay, so there are also biological doublets when we cannot biologically, separate two cells. For example, this might come from some solid tissues, right? There might be some cells which it's really difficult to break apart. And then what happens? We, we will see two cells in one droplet, and then we'll have, like, again, biological doublet. We would like to avoid them. So in order to avoid these kind of things, we can do, for example, nuclear single cell, right? When we do when we only sequence nucleus, right? There are some techniques, but we will not cover them. So nuclear single cell is basically you sequence only the nucleus, right? And as a result, if you have, for example, biological doublet, you can avoid this by nuclear single cell. Or again, like you can do it next in computational steps. Like if you see that some cells have suspiciously high gene like count of genes expressed, or for example overall number of UMIs. Like let's say on average we saw that, like from the previous pictures, right? On average we saw that we have, let's say, 5,000. 5,000 is quite a lot. Like let's say like 3,000 UMIs from a cell. If you see a cell which has, like, let's say 7,000 or 8,000, it means, okay, maybe there are like 3 cells. So you would like to remove this kind of cells.
Did you do it during the microfluidics? Use a camera to see if there's 2 cells and then shove the droplets out?
I never heard about this. I think this will be more expensive, right? Because this adds to the cost, this kind of things.
Currently, it's just like kind of like a dumb chip, right? Just flow stuff. It's like a very basic.
Yes.
Test.
Yes. Okay. Let's do a break for 5 minutes, and then we will switch to next topic. Okay. So let's see what's the output of single cell. It's basically the following. Is, let's say we have 2 cells, so cell 1 and cell 2, right? Let's look at these 2 cells. So what we can do, basically we can measure number of reads, right? And we can use, for example, a measure called PKM. So by the way, this blue and white ones are isoforms, so have different isoforms, right? And cell 1 and cell 2, and cell 1 is more expressed than cell 2, right? So basically, if we report just the raw read count, it doesn't tell us, like for example, we can see more reads from this cell, but it depends, on multiple things. Like we cannot directly compare 2 cells between themselves. So we need to somehow, normalize the counts so that we can compare, first of all, the genes inside of one cell, because for example, a gene might be longer, right? And as a result, like just statistically produces more reads. So that's why we need to account for the read length. So here, like in one of the approaches to measure gene expression, we use RPKM or FPKM. So FPKM stands for, RPKM is reads per kilobase of exon per million reads snapped, right? But FPKM is, instead of reads, we use fragments. Right? So basically what we do here, we take the number of reads or read count, normalize it by the length of the reads. So in this way we get RPK. All right, and then we also need to normalize here by the total sum, right? So this will be a number basically from 0 to 1, like a frequency, right? But we multiply them by 10 to the power 6, for example, right? So after this step, what we get that for each cell, let's say the total gene expression is 1 million, right? So we basically divide, we normalize the gene count by the length test, right? And then use this number to basically, to divide to the total number of reads. So this is one of the approaches to measure gene expression, but the other one is basically computing TPM. So TPM is transcripts per millions of reads, right? So here in TPM, what we do, we take this RPK and we basically multiply them by 1 million and divide over all other are RPKs. Okay, so basically, what we do here is we divide read counts by the length of the reads, right? And then basically divide over the total sum of such numbers for all the genes. So it's also like, as a result, we will have, each cell will have 1 million total expression. Okay, let's look at some examples here. All right, so how we compute these values, RPKM and TPM, right? Let's say we have here a table, and in this table we have the length of the genes. Let's say some gene is 2 kilobases, some other ones are longer and they have 10 kilobases, right? And we have 3 cells. And here each number is read count, right? For example, cell 1 produces 20 reads from gene A, which is 2 kilobases long, right? So, okay, we cannot directly compare, for example, the expression of, let's say, different genes, right, inside of one cell, because we know that they differ in length, and we need to account for this, right? And also, we cannot really compare the cells between themselves, because, like the total count right now is different. It depends on the cell, right? So we need to somehow, come up with some numbers which will be comparable across cells and genes. That's why we use this to measure. So, for example, in RPKM, what we do, we first normalize by the depth. So basically, what we do, we divide this number by. Is it the end of the class? We first divide, basically, we normalize by the depth, right? We divide 20 by 2. And then what happens next is that we normalize by the gene size, right? Or gene length. So, okay, and here in TPM, it's kind of a bit different. So we first normalize by gene size, and then we normalize by depth. So, for example, let's look at this one, right? So we have cell 1, we normalize by gene size. What happens is that here, let's take an example. So let's take cell 1, which has 20, 40, 10, and 5. Right? So when we account for gene length, or like how it's written here, gene size, we first have to divide it by the length of the gene. So, okay, as a result, what we see here, instead of 20, we first will see how much? 10. We need to divide this 20 by 10, by 2, and we will see 10. Then 40, we have to divide it by 4, and then we will see 10, right? Then 10, we have to divide by how much? By 1, right? We will see 10. And then basically 5, we'll have to divide by 10, right? This will be 0.5. Okay. Then what we should do here is basically we have to add up all these numbers, right? This will be how much? 30.5, right? This is basically the total gene expression adjusted for the length. And then, like if we want to have sum to be, let's say, 10, right? Then what happens? We divide this 10 by 30.5, right? And multiply it by 10. And then here will be 10 divided by 30.5 multiplied by 10. 10 divided by 30.5 multiplied by 10. And here will be 0.5 divided by 30.5 multiplied by 10. So when you do this kind of operation, what will happen? We will see that the total sum will be 10. This is, this depends on this 10 when we multiply. If we want to have total count 100, we multiply by 100, right? But basically, yeah, this is like, for example, how we produce TPMs. Right? Yeah, take the values from here, divide by the gene length, and then see what's the proportion of each gene. So this would be TPM. Okay, this was an exercise. let's keep it. So let's keep it. So this is basically the idea of how to measure gene expression normalized by gene length and depth, right? Because that's what we want. We want, again, like the same depth across different cells, and we also have to take care of different gene lengths. This is in the case of SmartSeq too, right? Because, for example, we have reads from all the transcript, right? And that's why we have to normalize by the gene length.
What does it matter the order that you do the normalizing by gene and then backwards?
It's just yeah. If you do the first one, you'll end up with RPKMs, right? And the second one, TPMs. So I think units are different, exactly, yes. But for example, in TPMs, what can happen if you see that the sum is the same across all the cells? So this will be more comparable between cells. So RPKM is basically, it's more like frequency of a gene inside of a cell. So you can compare relative abundance of genes inside of one cell, but it's not really comparable because you see, like, here is 4.07, 4.50. You cannot directly compare between cells. So okay, I think now people use more like TPMs, right? But in fact, like in 10x, it doesn't really matter. It's only read count because you don't have gene lengths. You have only 3' end. You don't normalize, right, by gene length. So these all things refer, to like SmartSeq protocols, or like full length. For 10x, we don't normalize for gene size, but what we normalize for the depth. So what can happen is the following: you have a cell in SmartSeq. We like the information about gene length is irrelevant. We don't need it, but we still need to somehow come up with some numbers which are comparable. So then what's the first step is you find the total expression, like the total read count. You divide each of them, and you find the proportion of each, or abundance of each, relative abundance of each gene. And then you do all of the steps, which we discuss next. So next would be normalization. Okay. So normalization is basically what do we do here? We want the data to look more normal, right? So what happens with these approaches here, let's say with TPM, right, is that like if you look at this example here, you will see that some of the genes have high number, high expression here, and some of them are really low, And what happens is that when you do any analysis from this kind of values, you will see that your analysis, all the results will be driven mostly by this highly expressed genes. So you like these genes will be kind of, like for example, if you want to visualize, or if you want to do maybe some like association or like many things, you will see that like most of the results will be explained by this highly expressed genes. And moreover, if you look at the gene expression, right, you will see that basically the genes, like for example here, right, or here. Okay, obviously they're not the same expressed across different cells, but the expression will not look like a normal distribution. So what we want to have, distribution of gene expression to look like normal, so that we can apply many methods. So why do we do this? For example, we want to remove, like, let's say, amplification bias, right? Or like remove the impact of highly expressed genes, right? So that's what we do. We, for example, scale by the library size. What it means basically the second step of TPM. So normalizing by depth is basically the same in this context as scale by the library size, right? Let's say one cell has 1,000 total count, and second cell has 5,000. You cannot directly compare the cells. So therefore, first step would be dividing the counts by the total library size, right? And then to make data look like more normal, what people usually do, they log normalize. it's called log normalization, but what is in fact is the fact that, for example, you get a number here, right? Like, let's say we had in that example 3.2, right? So, okay, what we do with this number, we take the log. So it's basically log 1 plus x. Why do we need to add here 1 is because sometimes when we have 0, we cannot take log of 0. It does not exist, right? So therefore, we take this log. And what happens, like our numbers look more or less coming from kind of fixed range. So for example, in TPM, when we multiply by 1 million, right? So here, when we multiply by 1 million, 10 to the power 6, like the total library size is 10 to the power 6. But what happens is that you will see some genes which have count, let's say, 10,000, and some genes which have count like 5. 10,000 and 5 is basically, it's a big difference. So we need to somehow make the genes not to be so much different from each other. So when we take log of 10,000, it will, it will be like much smaller number. Like, for example, if you take log base 10 of 100, it will be just 2, right? 1,000 will be 3. So as a result, instead of your numbers having like very high variance, you kind of stabilize them by, taking the log scale of expression. So as a result, like a gene which has like, for example, how much? 10,000, right? If you take log 10, it will be only 4 instead of 10,000. Okay. Next. So, okay, we know how to measure gene expression. What's the next step is visualization. So how to visualize the data? And basically, let's think that, for example, if we are dealing with human cells, we have, let's say 20,000 genes, right? So how can we visualize such data? If we had only 2 genes in each cell, it would be simple. We could come up with simple plots. Something like this one, where we would have gene 1 versus gene 2, right? And this would be simple. Each dot would be a cell, and we could look at our cells and do some conclusions, do many analysis. But in case when we have 20,000 dimensions or 20,000 genes, we cannot directly visualize it like this, right? Because we can, view or like fill up to 3 dimensions. So we need to somehow, transform our data or transform, for example, the coordinates, so that basically we view our data as two-dimensional instead of 20,000 dimensional, right? So usual statistical approach for this is principal component analysis. Okay, I assume that some of you should be familiar with this. So basically, is a transformation of coordinates such that we basically do, so we transform the coordinates in such a way that our axis, like for example, the first axis which we choose, it basically goes along the direction which is the most variable, right? What I mean by this is the following. Let's say we have, okay, let's have like some cells, right? And our data is somehow like this. So we see that in this direction, for example, gene 2 is more variable, right? Or let's make it not like this, but somehow, let's say a cluster of cells like this. Assume that we have more directions, but here, okay, I'm trying to give a simpler example. So if you have that we have a bunch of cells and neither of gene 2 or gene 1 are kind of expressed, but they're a combination, right? So if you look at along this line, we'll see that this line explains the most variance of the data, right? Because along this direction, my data varies much more than along, for example, gene 1 or gene 2 or gene 1 direction, right? So this is the idea of PCA. We transform the coordinates. In this case, we would just use this as the first coordinate, and then, for example, this one would be the second coordinate. right? You transform linearly your coordinates such that your new axis or new principal components, they explain most variance, right? And this new coordinates is just, they are just linear combinations of your genes. Why linear combination? Because we just take the, for example, we take this real axis where we have gene 1 and gene 2, and we just somehow transform, like for example, this will be some shift and then some rotation, right? This would be linear combination of gene 1 and gene 2. So this axis. So this is another visualization. I don't know if it's much clearer than what I'm trying to explain here. But this is already like in 3D, right? We have gene 1, gene 2, and gene 3. And then there exists, you know, some, let's say some plane, right? Such that when we project all the cells which have 3 genes, when we project them onto this plane, right? So one of the directions which basically explains a lot of the variance, for the, you know, the largest variance is along this line. It will be principal component 1, or the best coordinate, and then the second will be orthogonal to this one. So this will be the principal component 2. And when we project the cells, obviously we lose some information, right? For example, here we have like all the cells in 3D, and here we see that all the cells are in 2D. But basically, it allows us, you know, to visualize like in 2D instead of 3D, and it also applies to, like, you know, any number of dimensions. If you have 20,000 of dimensions, you still can project two most highly variable, you know, dimensions. So this will be principal component 1 and 2, and if your cell types, assume that you know, these symbols correspond to different cell types, you know, like sometimes you can visually see that your cell types are separable. If they're really like separable, you know, like for example, if they're separable in like in 20,000 dimensions, there is a chance that they will be separable in 2D, but not necessarily. You know, there might be, for example, like two cell types which are somehow, you know, like for example, the red one will be here on this side of the plane, and then some other one will be exactly in that place, but on the opposite side of the plane. And they can be projected into line bridges. So you can visualize different cell types, but not necessarily in all the cases you will see a good separation between cell types. Okay. There's another approach which can basically avoid this issue, you know, of different cell types mixing in a plot, which is called t-SNE plot. So t-SNE stands for t-distributed neighborhood embedding, something like that. I don't remember the whole abbreviation, but the idea is that PCA is a linear transformation, right? And it means that it kind of preserves the, you know, the structure of the data set in a sense. But what happens if the two cell types are not linearly separable? It means that not in all the cases with PCA, we will be able to view them as different clusters. So t-SNE is a kind of, also is a dimensionality reduction technique, but it's non-linear. So non-linear means that, you know, basically we don't know which, so what each of the axes represent in this case. So in PCA, there is drawback that we cannot see all the time separability of cell types, but at least there is, you know, interpretability of axes. So we know that principal component 1 is a linear combination of some genes, principal component 2 is linear combination of some other genes. With such technique as like t-SNE, t-distributed stochastic neighborhood embedding, not, you know, you don't have clear explanation of what each axis is. But what happens is that the idea is the following: if you see that if your two cells are close in 20,000 dimensional space, when you do this t-SNE plot, they will be close here in 2D. So basically, it preserves, some kind of local structure of your data set. So this is a good tool for visualizing clusters. You cannot do anything else. Like sometimes people use this as dimensionality reduction, but it's not really meaningful. It's only used for visualization. Because closed cells in 20,000 D, they will be closed in 2D here. Yes.
So, I mean, well, yeah. What?
What's the interpretation of this?
There's no. When we're saying it's gobbledygook, right? There's no intrinsic meaning to these things, right?
Yes.
That also makes it non-determinative, right? So.
What it means by none?
Because I have three-dimensional space. I got three objects in three-dimensional space. They're a certain distance apart from each other, and I'm going to try to say that these two things are 5 apart, and these two things are 7 apart, or something. And then I put them together onto a two-dimensional thing. They cannot fit together. You have to fudge the distance between them, right?
Yeah, I think so.
Otherwise, you'd be describing a 20,000 dimensional object. So you have to fudge. You have to kind of fudge the distance between two purple dots is not the actual distance in 20,000 dimensional space.
No, it's just the relationship between distances is kept in a sense. There is also like MDS, multi-dimensional scaling, which is another transformation, linear transformation, which basically you try to fit the objects to embed the cells into two-dimensional space, but keeping the distances between pairwise distances. There's MDS.
Pairwise.
But here you don't keep anything. The only thing you keep is that closeness of cells. That's the only thing which remains. So if the cells are close in 20,000 D, they will be close here in the plot. But you cannot really use this coordinates from this plot, right? You cannot really use these pictures for clustering actions because there is no interpretation of what are the coordinates. Like in case of PCA, at least there is some interpretability that this is some kind of meta gene, or combination of genes.
Yeah.
In this case is
because the PCA could be represented as a vector with all of your genes. Like, oh, this PC is mostly gene XYZ.
Yes.
Right. That's
exactly.
Contributor to this piece.
Yes.
They are different clusters.
Yeah, that's it. So we can only visualize clusters.
Yeah, it's like this Ouroboros.
So we'll produce this kind of plots tomorrow in the class.
That with clusters, you can go deeper and say which type cells and.
No, not really. Okay, we'll go to this. Okay, cell type identification. You are anticipating my slides. So what is a cell type? No. Okay, for biologists is what is a group of morphologically similar cells, right? The biologist looks at the cell, finds some cell proteins, some kind of morphological things, and then can distinguish between different cell types, right? So definition in single cell RNA-seq is kind of different. So basically, these are cells which have similar expression patterns, right? So what it means that when we look at the plots like TC, so okay, when we see that, for example, two cells, if they're close to each other, then they probably have the same expression patterns, right? And then we can group them into the same cell type. So how to define cell type? Okay, and I have slides on clustering, but let's first talk about how to cluster cells into different groups, and then how to mark or how to label cell types. Okay, so for example, one of the approaches is the most standard one is to have your cells, and then you can represent a cell as a vector in multidimensional space where your dimensions will be, for example, genes, right? K-means is a good algorithm that is kind of scalable, and you don't necessarily need to reduce the number of dimensions. You can work with 20,000 dimensions with K-means, right? So what would be the output? The output would be kind of clustering, or like, you split your cells into groups. Like, for example, in this picture, we'll see that we have 3 clusters here, right? This is the way how k-means is producing your clusters, and then you assume that each cluster here is a separate cell type. Now, the question would be how to understand what cell type is this group of cells. So this is a different question. So, okay, you should do also some next steps. Like, for example, you should identify which genes are more expressed in this group of cells as compared to other group of cells. So these genes are called marker genes. And then if you know that, for example, let's say PBMC cells, right? Like if you're dealing with B cells, you know that MS4A1 gene is upregulated in B cells, right? And then if you found that, for example, a cluster where you have this gene highly expressed, then you assume that this cluster is B cells. That's how you do it. But here we are talking about different ways of clustering, right? So in k-means, the main idea is that, okay, it's really scalable, but the problem is that you need to give the number of clusters. Like, let's say if you're working, let's say with some, let's say retina tissue, right? You would assume that there are like 30 clusters. So this many clusters you would find with k-means. So you need to provide this parameter. Otherwise, it doesn't know how many clusters there you have there.
But if you isolate like a certain type of cell, like a invariant cell, then you have only one cluster, you have only one certain type, because you fast them, so you sorted them only that.
But you have subtypes.
You have subtypes, correct?
Subtypes.
Exactly. But then like, for example, with k-means, right? Even if you provide the cells of the same cell type, right? Like any type of clustering will anyway split the data. There is no clustering which will say, no, you should not split, there is only one cluster. You say, okay, give me 3 clusters here. It will split it.
So you're saying that if you do k-means clustering, you force the number of clusters.
Yes, you should choose them
The best.
in advance, right?
Cluster by defined by k-means, you can see that usually are very well correlating with
With TC, usually how we do, and we will do tomorrow. So what we'll do tomorrow, we will first cluster cells and then visualize in TC. So I think that if your clustering is good enough, then when you color your cells by cell type label or by cluster label, they will be more or less separated. Sometimes it happens when your clustering is not good. You see that like TC shows you separate groups of cells, but some groups of cells will have different colors. Okay, there are all kinds of clusterings, like for example, hierarchical clustering, right? This means the following, that we start, like we have some cells, right, input cells, and then what happens, we like iteratively we look at, for example, the most similar cells, like B and C is the most similar cell, so that's why we kind of split them into, we group them into one cluster. D and E are also similar, so we end up with 2 clusters, and then, for example, we look at cell F, which is, closest to DE than to any other ones. So we, we increase the size of this cluster to 1. So as a result, we have 3. And then we see that DC and DF are more similar in some of the metrics. Like, for example, it might be equivalent distance or any other similarity measure. If you see that, for example, 2 clusters are more similar to each other than DF to A and DC to A, you merge them, and then the last one you have like all the cells. So here, this kind of hierarchical clustering, it gives you like multi-level clustering, so you can decide, for example, at each level to split the cells. But again, how do you decide it? It depends maybe on your biological interpretations. Like where to split. If you split it here, you have only 1 cluster, right? If you decide to have 2 clusters, you should say, okay, I have like 1 cluster here, and everything else is the second cluster. This is kind of a tree approach. We start with 1 cluster, and it depends on where you split it. There are also all kind of, for example, graph clustering. I think this is the most often used approach in single cell. So basically, each cell is assumed to be a node or vertex in a graph, right? And then we have, for example, edges. It means that, for example, these two cells are kind of similar to each other, right? And the weight of each edge is, let's say, distance between them. So what we would like to do is to split this graph into, if you know what is a clique. Clique is, a group of cells which are highly similar to each other. So this is another type of approaches. We don't directly do this in the software when we analyze the cells, but this is just for us, to know what kind of approaches is this. So next, when you do cell type classification, or like cell type, basically when you do clustering, after clustering you need to do differential gene expression, right? So here is the following. Okay, this is a simple illustration. Let's say we have cell type A and cell type B, right? There will be some genes which are kind of stable across the cell types. They will have similar gene expression. So let's look at this genes here. For example, AFTPH and this one, they have kind of similar gene expression across different cell types, right? But for example, part 3 gene, it has, like, let's say the average count is how much? 5/3, 3 + 2 divided by 3. And this one has only 2/3, right? So it's basically, we see that this one, it has, like approximately twice larger average gene expression. So in differential gene expression, we identify this kind of genes, which are highly expressed in one cell type and not expressed in the other one. How it's done is usually, you apply some statistical methods in order to select, highly expressed genes by, statistical significance and log-fold change between two conditions or between two cell types, as in this case. And it's not only, just ratio of two genes, which might be, used in like some simple settings. Okay, software tools. So there are multiple tools which, which we can use for different stages of the analysis in single cell. So for example, when we align reads, there are multiple alignments, for example, star and HiSat2. So this star is a part of 10x pipeline. So 10x has their own software. They use, for example, star, right? Again, like if you're dealing with SmartSeq data, you can quantify gene expression by using Callisto, right? For example, differential gene expression between different cell types, we're using Seurat, and that's one of the, this is the main tool, I would say, in single cell. Usually like people, like I don't know, 99% of people who are using R, they're using Seurat for single cell analysis. Because Seurat is basically, it can do a lot of things here. Like it can, cluster genes, cluster cells, right? It can do differential gene expression. It can do many things. So Seurat will be one of the, it's one of the main tools which allows you, to analyze, let's say, 10x datasets or SmartSeq2 datasets in, just in minutes. It's very easy. Tomorrow we'll use Seurat. Sorry? Seurat script was Satija Lab from New York. What also?
Not many they have like most of the programs to analyze the data by their device.
Oh no. Seurat it's not like, okay, 10x, and this is a good observation. So 10x, because it's it's a like commercial company, right? What they do, they have their own software tool which is used to for analyzing their data. So okay, they have some raw data files, but usually we're not using those ones. So what happens, you run like it's called Cell Ranger, their software. So you're using Cell Ranger. So Cell Ranger is a suite of tools from 10x, which basically produces you the gene by cell matrix. All right, and then in Seurat, what we do, we load the gene by cell matrix, and then we do all kind of nice things. We find variable genes, we filter out some bad cells. You know, we can visualize cluster cells, find differential expression between clusters. So Seurat is basically, it's working from gene expression matrix, right?
So does this mean that I can do the alignment using Syranger and then take whatever the output they get from the alignment to the star alignment from Syranger and put into the Seurat? And
Yes, but CellRanger will do this all for you. So CellRanger inside has star.
okay.
If you download, have you ever downloaded?
Yes.
You will see there is a folder star. So it has inside this star. Otherwise, if you're like, I'm talking, you know, in general, not specifically binding to 10x, but for example, you might have SmartSeq2 or any other technology. In this case, you would need to use these steps by yourself. So when I talked about SmartSeq2 and I said that, okay, it's easy to use open source, it means that you can use all these tools, you know, you can use a combination of tools, but 10x, they have, okay, although they use like Star, which is kind of open source, right? But they have like some, you know, some customized pipeline for it. Oh, okay. Okay. Yeah, but now okay, fine. So anyway, if you, for example, if you want to do, let's say, cell trajectory analysis, right, or like pseudo-time analysis, this feature is missing in Seurat, but you can install some other tools, like for example, Monocle. There is another tool. So all kind of tools is all available on Bioconductor, our packages. So you know, people who are using single cell, they're mostly using R, right? And a lot of packages, like Bioconductor has thousands of packages there for all types of analysis. But some people use Python, and there is, for example, ScanPy. So ScanPy is kind of Seurat, which is in Python, you know. It depends on what you're using, but most people use Seurat. For example, I'm more, you know, using Python, but because I needed, you know, like some good tool, and Seurat is a good tool, that's why I'm using R, just because of Seurat.
If you want to like talk to each other, well, let's say you do some stuff in Seurat, and you're like, okay, so this I want to do some.
Oh yeah, it's possible. In R, you mean R and Python?
Well,
talking to
to do like pseudo-time analysis.
Uh-huh.
You want to use Bioconductor, but most other things use Seurat. So what's the order in which you should do things?
Yeah, so
you count up your stuff, then you do something in Seurat, then you do something in Bioconductor, you export.
yeah, so Seurat is just a library which you import, like you install and import in R. The same like other libraries from Bioconductor, like Monocle or any other libraries, you can just import and work directly with them. It depends if they're compatible with Seurat, because most of the single cell packages, they're compatible with Seurat.
Okay.
So there is, for example, even like a function, for example, in Monocle, which takes a Seurat object and works with it, you know, because they're all based on some other packages. It's a huge number of packages which depend on each other.
So you can pass a Seurat object to Bioconductor. You can do
Yeah.
You can be like doing Seurat stuff and then pass it.
Yeah, in case, for example, if this doesn't, yes, in case if this doesn't, if it doesn't work, then you just extract your data matrix from Seurat object and you supply it to other methods or like other packages. It's even possible, you know, to have a communication between Python and R. So, for example, you can have some Python objects and you want to import it into R. There are also libraries for doing this. We'll not go into this ones, but what we'll do tomorrow is Seurat. We just need some basic analysis. So, okay, I think we're approaching to the end. So here, let's see the following. We have multiple single cell data sets, right? And here are some links which go to, let's say, like some what is brain data sets, right? There are multiple, like for example, pancreas data sets. Like for example, pancreas data sets, we can find them on Seurat webpage, and they consist of like 8 different technologies. You can like view them, do all kind of analysis, right? There are a lot of data sets from 10x Genomics webpage. You go there, you will see a lot of PBMC, like mouse cells, 8,000 cells, 68, a lot of data sets. So you can, for example, analyze these data sets and like come up with some algorithms for single cell data. Okay. The last thing to cover here is when we want to do some single cell experiments, what things should we consider in order to make it more efficient. So first thing is what experimental design, how we process reads, right? How we prepare gene expression matrix, and how we interpret the data biologically. So these are like main things to consider here. And for example, for experimental design, this is very important here because as I was telling in the beginning, you cannot sequence, like multiple cells, or if you have, let's say, like I have a data set from our collaborators which consists of 700,000 cells coming from 160 individuals. You cannot sequence them at once so that there is no technical differences between the data sets. So you need to split your data sets into different batches, right? So basically, you should avoid confounding of biological and batch effects. So what it means the following. Let's say you have 2 samples, 2 individuals. One is, let's say, healthy, like case, and the other one is control, right? Not okay. Let's not talk about any specific diseases. One is case, another one is control. So what happens? You would like to like see, for example, what's the difference between some gene expression in some cells or some cell type, right? And you have a lot of cells from both of them. So what can happen is that like if you're not careful designing your experiment, you would do the following. Let's process in one run one sample, and then let's process in another one the second sample. And so what happens here in this case, you are confounding your biological signal with technical effects, right? So what happens, you will see some differences between 2 samples when they're processed in multiple runs. But this difference, it might come, you will not be able basically to distinguish, because you will confound biological and technical effects. So here, for example, if you have a case of multiple biological conditions, so your conditions should be captured on the same chip if possible. So it means that on the same chip, you should basically design such an experiment so that on the same chip, you will have both case and controls, right? Cases and controls should be there so that you will not be, confounded by technical effects.
Yeah. So you have like different source and then disease cases. Then you should run for each one control and one disease. No?
It depends on how many samples you can fit on one chip, for example.
Say
Yeah, I think
this one, maybe you sort different type cells and then you have sequence.
yes, or you can come up with a more complicated, like for example, there's less number of cells, like you can split one sample into multiple, and then compare across batches. Correct.
But then I will compare control and case. If I will submit 10 times, one control in one case, then I will compare between control and case, or I should sum all 10 and all the other 10.
Yeah, that's kind of another problem. For example, if you have 5 controls and 5 cases and you split them, one control and one case go to 5 different chips, then probably you will see also differences between your cases as well as differences between your controls, right? So you need somehow to split the cells. You need to have a chip which will have multiple cases and multiple controls. You understand what I mean? So it's if you have, for example, one chip here, basically, okay, like one chip here, you have case one and control one, right? And then you have the second chip, you have case two and control two. What can happen is that your cases will have, also technical differences between them. So there might be also biologically different, right? Cases. For example, you're comparing some disease versus kind of healthy, but maybe these healthy people have some other disease. It's kind of difficult. So that's why here in this pictures you see that you should have unconfounded ones. So you have biological condition 1, 2, and 3, right? And you put them all into the same chip. So so if they will have some bias, they will have it all. That's the main idea here. So what kind of chip? It's one of these chip technologies. I'm not sure exactly which technology is this one. So otherwise, for example, if you see some batch effects, like you might have some, some confounding still in your experience. So there are tools which are designed for removing batch effects. Okay. So I'm doing such a tool, but it's not yet ready to be advertised. Okay. It will be, yeah. It's already on GitHub, but it's still in development. I'm still developing such a tool. So okay, you should also, like, for example, consider processing rates, right? Let's say if you're dealing with SmartSeq, so you'll need to do all the steps like alignment by yourself, as compared to 10x, which has a whole pipeline there. So you'll need to, for example, trim your reads, right? You need to map your reads, right? For example, you need what for small data sets or UMI data sets, you need to align to genome using star, right? For larger data sets, star is not the best choice because it's very slow as compared to, for example, tools like Callisto, right? For example, for large data sets, you'll need to use Callisto. And if you want to quantify, right, quantify gene expression, then for some, it also depends on, for example, the size of the data sets or whether you have UMIs or not, you will need to use different tools, right? So how to prepare gene expression matrix? Also, you should, consider, for example, you can control your experiment by, for example, some spike-ins. So spike-in is basically some particular type of RNA which you kind of mix into your RNA, and you know how much to expect this spike-in, right? So if you see that your spike-ins is somehow biased, then probably there is some problem, and you can adjust gene expression by using the spike-ins across different samples, right? Okay, you can use, for example, different tools for library size normalization, because we consider it here in this workshop only, like dividing cell read counts by the total library size, but this might be, not a good idea for some cases. I don't know, like some statistical analysis which like SCRAN, they can produce some other type of normalization. Or for example, for batch correction, if you have, again, multiple experiments, use tools. Yeah, out of these two, for example, I would advise you to use Combat rather than this one, eminent correct. But it depends, it depends on which, what you have. You have a balanced design. Yes, you rather use this tool because it's used, in some fitting some probability distributions, and that's why it's really important to have balanced design. If it's unbalanced design, if you don't know, for example, cell types, right, then you should use. No, they are already developed. I have my tool which I'm developing, which I'm not yet advertising here, but hopefully, if I will get some success. Next. Spike-ins is basically a specific RNA, type of RNA, which you already know, like the sequence of that RNA. You incorporate it in your sample, so that when you sequence, you will know how much expression this spike-in will have. So this is used for kind of controlling your experiment. This is spiking. Okay, for biological interpretation, there are multiple tools. For example, for feature selection, there are feature selection means extracting, for example, some useful genes or like meaningful genes, right? You can use M3DROP, for example, for clustering Seurat. For differential expression, I would also use Seurat, right? But there are multiple other things which, for example, depend on how balanced your batches are, right? So I would use also, let's say, Seurat, or you can use EdgeR, right? In some cases. So for pseudotime, we cannot use Seurat because it does not have this feature, but we can use, for example, Monocle. This is the tool of choice in most of the cases for pseudotime analysis. So there are like a whole bunch of tools. What we will talk about tomorrow is Seurat. So that's why my advice to you guys is to try to install Seurat tomorrow. I'll email you about like where to find, like you can Google, but fine, I cannot tell you just go and Google. I will send you an email with the which will point you to the download page and how to install Seurat into RStudio. So that's why when you come tomorrow, I'm expecting you to have everything working. If not, we will spend some 10 minutes, No, RStudio doesn't have. So it's just with Seurat, I think I don't remember. It's either on Biocontact or you should just install packages Seurat, something like that. It should be simple enough, but I'm expecting you to have, for example, R of version 3.5 or 3.6, right? If you have this version, so then you'll be able to have the latest version, because Seurat comes with multiple versions. So right now it's version 3.1 something like, or 3.2. You had a question?
I was wondering what you meant by balance versus unbalanced batches.
For example,
Multiple samples.
this might be one of the things, or how many cells you have with different batches. If you have like one batch, like 100 cells, and the other one 10,000 is.
So it's similar.
Yeah. Okay. Thank you for attention. That's all for today. But tomorrow, yeah, please. Oh, yeah. By the way, that's all with the slides. But I would like you guys to have another pass on the test. So let's please go to this one. I just want to make sure that that this class is kind of useful. Okay, it's short. I agree, but by tomorrow you can maybe like I will send you the slides, or you will go and Google many things. But let's see, let's see how it goes here. So don't be too much stressed if you like don't remember something. It's okay. But in case if you are a graduate student, so okay, so I would advise you, to be more serious with this, because we will need to probably like come up with some grading because this is a class 275B, right? So if you're a graduate student, so try to attend. So this is very important. If you're a graduate student, you should attend these classes, and it's very important for us to see that, for example, you can solve this quiz. It's very simple, just 10 questions, multiple choice. Done? Okay. Yeah, I think tomorrow we'll have more interesting class because it's just talking today, and tomorrow we'll do more interactive session. Okay, yeah, I think that's it. So see you tomorrow.